So another thing that you do need to understand about the antibiotics is how they work. So their mechanism of action is what they actually do to inhibit the bacteria from growing. And this is also something that you will need to discuss in the discussion part of your lab report. So every antibiotic has a different mechanism of action. So chloramphenicol is an antibiotic that prevents peptide bond formation during translation. So you may or may not remember um, replication, transcription, and translation. If you don't, we're doing this in chapter 8 in lecture. But translation is the process of producing a protein, and peptide bond formation is basically the amino acids binding together to form that protein. So chloramphenicol inhibits peptide bond formation during translation, which really means that it stops the synthesis of protein. If bacteria cannot produce protein, they cannot grow and divide, and they do die off. Ciprofloxacin, or Cipro, it's an antibiotic that interferes with DNA replication. Bacterial cells have to replicate their DNA to give it to additional daughter cells when they go through binary fission or growth cycles. So if they can't replicate their DNA, they can't produce more bacterial cells, and then they die off. Penicillin. Penicillin inhibits the cross-linking of peptidoglycan in the cell wall. Bacterial cells need peptidoglycan in their cell wall. That peptidoglycan in their cell wall gives the cell kind of shape, structure, and support. And if it's destroyed in any way, what happens is it causes the bacterial cells to burst through cell lysis. The last one, trimethoprim. Trimethoprim inhibits purine and pyrimidine synthesis. We'll talk more about purines and pyrimidines in chapter 8, but purines and pyrimidines are nitrogenous bases that are used in DNA and in RNA. You might vaguely remember the A's, the G's, the T's, the C's, and the U's, like adenine and guanine, uracil. Those are nitrogenous bases. But by inhibiting the synthesis of purines and pyrimidines, you really shut down major cellular processes like replication, transcription, and translation, and inevitably cells are not able to survive without purines and pyrimidines. The other little things on here, this 30 micrograms, 5 micrograms, 10 units, and 5 micrograms, these are the dosages, how much of the antibiotic is present in that little disc, which is something that's important for the procedure section of your lab report. And then these are just the codes as well. So bacteria also have resistance mechanisms. So antibiotics have mechanisms of action. Antibiotics have a way to kill bacteria. But unfortunately, bacteria have a way to become resistant, which is a way that they can learn to survive and tolerate the drug. So even though you're taking the drug, it has no effect on bacteria. So drug resistance is an adaptive response. It's kind of a learned response where microorganisms begin to tolerate a drug that normally would have been inhibitory, but now they're able to survive in the presence of it. So it's quite a bad thing. So what are some things that bacteria can do to become resistant to an antibiotic? This is something, again, that you would need to mention in the discussion portion of your lab report. Some antibiotics just don't uptake the drug. A lot of those drugs, like chloramphenicol and Cipro, they have to get inside of the cell in order to work on things like replication or translation. So the cell simply just doesn't absorb the medication. So 3 of the 4 medications that we're going to be working with for our Kirby Bauer are known to have poor uptake with certain strains of bacteria. That's C30, CIP5, and then P10. There's also inactivation of the drug. Some bacteria can produce a chemical that deactivates the antibiotic. The most famous one is the production of beta-lactamase. A lot of bacteria nowadays produce a chemical called beta-lactamase. Beta-lactamase is a chemical that can inhibit penicillin from working. So let's say you're infected with some type of Staphylococcus bacteria and you're given penicillin to cure that infection. There's a pretty good chance that that Staphylococcus bacteria produces this beta-lactamase enzyme. That enzyme is actually going to destroy the penicillin as soon as you start taking it. So that medication will not be effective at all because those bacteria are completely resistant to penicillin. So penicillin, a lot of times bacteria are resistant to it because of drug inactivation through beta-lactamase C30, or chloramphenicol. It has shown to have some resistance as well from bacteria producing some type of chemical to inactivate it. There's also altered target. Penicillin, Cipro, and trimethoprim have all been shown to be resistant in regards to certain bacteria because of altered target. So an example of this is sometimes bacteria can change the way that they look so that the antibiotic doesn't recognize what it's supposed to be targeting. So if an antibiotic is supposed to target protein synthesis and the ribosomes, well, the bacteria can change the way its ribosomes look so that the antibiotic no longer recognizes it. Or if the antibiotic is targeting the cell wall, well, the bacteria can change the way that their cell wall looks so that the antibiotic can no longer target it. Or if the antibiotic is targeting DNA replication, it can change kind of the way the DNA appears. So again, the bacteria will become resistant to the antibiotic. The last one is one that we don't see with our certain antibiotics that we're working with, but it still is out there. It's called an efflux mechanism. Basically what happens is the antibiotic does go into the cell, but then the bacteria pump it right back out. So they kind of recognize like, oh, this is a bad chemical, and they pump it right back out of their cell membrane so that the antibiotic cannot affect them, and that's called an efflux mechanism. So it's definitely important that you understand how these antibiotics work, but also what the bacteria can do to become resistant to the antibiotic. So there is a procedure video that I want you to watch in D2L because when you write your lab report, you're going to have to write a procedure section. So I want you to watch the video to see how a Kirby-Bauer test is done from the beginning to end so that you'll be able to draft a procedure for your lab report. But that's in D2L. As far as your to-do list, you do again need to watch that procedure video. I want you to perform the Kirby-Bauer virtual lab. There is a folder of documents that I want you to look at for your lab report. So there's instructions for the lab report, there's some voiceover videos for the lab report, and it's also some additional resources that I want you to look at. And then of course take your week 5 lab quiz. So to kind of look at some of the things in D2L, so under lab week 5, you have your PowerPoint, the voiceover videos for the PowerPoint. This folder is your lab report resources. So there's a lot in here that you need to look at. Instructions for the lab report and the lab report rubric are here. Voiceover videos to explain the instructions for the lab report are here. This tutorial video is from our librarian to show you how to get access to resources to do the research for your lab report. And then this is a link to the LibGuide that's going to give you access to Bergies, which is like an encyclopedia of bacteria, and then also additional resources for doing your research on your lab report. Then we obviously have the week 5 quiz, and then your virtual Kirby-Bauer lab. And then there will be the procedure video, which I still need to post. So the virtual Kirby-Bauer lab, you'll click on it. If this pops up, that's normal. Hopefully you have the most recent Adobe Flash. You would just click here and then click Allow. And then you can read through this stuff if you want to. I'm just going to click Start just to get it started. So this doesn't show you the whole procedure for the Kirby-Bauer. It doesn't go through the whole part, but it does show you how to measure the zones of inhibition. So it's quite simple. All you do is take this roller and drag it up to measure the zone of inhibition. So we're kind of looking at this one, and it's like about 25 millimeters. So you want to measure it in millimeters. And then that is TM. So we see where TM is down here, which is right here. And we see where 25 falls. So if it's anything 12 and under, that means resistant. 13 to 14 is intermediate. And then anything greater than 15 is susceptible. So at 25, it is definitely in that susceptible range. So we click here and it lights up green. So that means it's right. If you would have clicked either of the other ones, it would have shown you it's wrong. So all you need to do is go through and measure all of these, and then you just click your answer and it'll tell you if you're right or wrong. These ones, like if we look here, there's no zone of inhibition. We just kind of assume that that's like 0 millimeters, nothing exists, and those are always kind of definitely in that resistance category. So make sure that you look at all the resources. I know there's a ton of stuff this week with the lab report coming up as well. And then just let me know if you have any questions as you go through everything.